today we are going to study the structure of DNA. In that we are going to study what is the repeating unit of DNA. So let's see. A repeating unit in DNA is known as, or a repeating unit of DNA is known as a nucleotide. A nucleotide. So what is a nucleotide? Basically, it contains deoxyribose sugar plus a nitrogenous base either adenine or guanine or thymine or cytosine plus a phosphate group. The phosphate group is denoted as H3PO4. It is denoted as H3PO4. When a nucleotide lacks a phosphate group, it is known as nucleoside. So basically, a nucleoside is made up of deoxyribose sugar plus nitrogenous base. There is no phosphate group. So which nitrogenous base? Either adenine or guanine or thymine or cytosine, one of these. Okay. So a nucleoside is made up of deoxyribose sugar plus a nitrogenous base, while a nucleotide is made up of deoxyribose sugar plus nitrogenous base plus a phosphate group. Okay. So now you know that what is a nucleotide and what is a nucleoside. Now let's see the different kinds of nitrogenous bases with their ultrastructure. So how many nitrogenous bases are there in DNA? 4 kinds of nitrogenous bases are there in DNA. So what are those 4 kinds of nitrogenous bases in DNA? Let's see. Those are known as, A stands for adenine, G stands for guanine, T stands for thymine, and C stands for cytosine. Out of these 4 bases, 2 are double ring structures. Known as purines. Purines. And 2 are single ring structures known as pyrimidines. Okay, so let's see how they are present and what is their ultrastructure. Purines are double ring structures and pyrimidines are single ring structures. Purines are denoted as Pu and pyrimidines are denoted as Py. So how many purines are there? There are 2 purines, Pu. Number of purines are adenine and guanine. And number of pyrimidines are also 2. Those are thymine and cytosine. Okay. The pairing in DNA is always purine pairing with pyrimidine or pyrimidine pairing with purine. So the pairing is always purine pyrimidine or pyrimidine purine. Now let's see the structure of purines. So how many purines are there in DNA? There are 2 purines in DNA. What are those? Number 1 is adenine and number 2 is guanine. So let's see their structures. Adenine is a double ring structure having 2 ring nitrogens. How many ring nitrogens? In the first ring, 2 ring nitrogens, and in the second ring it is having another 2 nitrogens. Okay, as you can see here, in the second ring the adenine is having another 2 nitrogens. So basically there are 4 nitrogen atoms. It is having some groups which are the only difference between two purines. So what are these? At this position it is amino group, at this position it is hydrogen atom. So let's complete the bonding. So the bonding is like this. As you can see, there is one hydrogen, there is one hydrogen. So now the bonding is complete. This is the structure of adenine. Now let's see the structure of guanine. Guanine contains NH2 at the bottom position. So nitrogen, nitrogen, then second ring, nitrogen, nitrogen, and the structure of guanine is complete. It contains ONH2 structure. So amino group is present at this position here. The ring nitrogen has hydrogen atom outside, and there is a double bond. This is the complete structure of guanine. So this is first purine, adenine, denoted as A, and this is second purine, guanine, denoted as G. Okay, so these are the two purines in DNA which are double ring structures. What are purines? Nitrogenous bases. Purines are nitrogenous bases. So if possible, draw it. I will continue with the pyrimidines now. Now let's continue with the structure of pyrimidines. So what are pyrimidines? Pyrimidines are single ring structures of nitrogenous bases. These are single ring structures. So how we can draw their structure? Let's see. So there are basically two pyrimidines. Number one is cytosine, denoted as C, and thymine, denoted as T. So these two are pyrimidine nitrogenous bases in DNA, denoted as C and T. So let's see the structure of C. The C contains 2 ring nitrogens. As it is a single ring structure, it contains only 2 nitrogens because there is only one ring. So here it is NH2 and here it is O. Double bonds are inside. Okay, so let's see, is it correct by valency? Yes, the structure is correct by valency. So it is cytosine. Now what is thymine? It contains at the position of both groups oxygen out of the ring. Oxygen atoms which are out of the ring. So let's see, double bond O, there is a CH3 and then H. So this is the structure of thymine. This is the structure of thymine. Okay, so this is thymine, denoted as T. This is cytosine, denoted as C. Okay, let's check the valencies. Yes, carbon 4 valencies, hydrogen 1, nitrogen 3, oxygen 4. Yes, all valencies are correct. After this, we are going to study about the numbering of these rings. Let's see how the numbering is given there according to the chemical nomenclature. Now let's see how the number is given to the purine molecule and to the pyrimidine molecule. Okay, so let's draw a purine molecule first along with the pyrimidine molecule. Okay, So it is purine and it is pyrimidine. Okay. Now let's see the purine molecule. Okay. The purine molecule is like this. It is having 4 nitrogens in the ring because it is having 2 rings. So let's see ring number 1, ring number 2. Ring number 1 contains 2 nitrogen, ring number 2 also contains 2 nitrogen. So let's see how it is numbered. The numbering starts from this nitrogen 1, 2, 3, 4, 5, 6, then 7, 8 and 9. Exactly different type of numbering is there in pyrimidine single ring structure. There are only 2 nitrogens because there is only 1 ring. Okay, so numbering starts from here. How? Let's see. Number 1, 2, 3, 4, 5. Likewise in clockwise direction. 1, 2, 3, 4, 5 and 6. This is the numbering of pyrimidine molecules. Okay. So this is enough for this video. We will meet you in another video. Till then, bye bye.